I'm Orion Venero. I'm in Kate Automala's lab at the University of Minnesota, and today I'm going to be talking about the kind of container side of Tyler's project. My talk is titled Contending with Containers, and it will really be about how we're going to build the liposomes to ultimately build the synthetic cells that Tyler was talking about. I think as biologists, we oftentimes think about the interesting biology going on inside of the synthetic cells, and we kind of take for granted the methodology used to build the liposomes, but certainly it needs some consideration in this regard. So I think just a quick note on definitions. For our purposes, liposomes and lipid particles are very different things, right? So lipid particles are something that retain their contents in a hydrophobic core. This could be for like drug delivery or various different applications. But a liposome really is something that has a really nicely defined bilayer and aqueous core. So here I'm showing you some like molecular simulations, starting from the bottom left and going to the top left, counterclockwise, from lipid particle more towards kind of the liposomes that we're interested in building. And if you think about like the COVID vaccine and its success, I think that you would be remiss to think that liposomes and forming them is pretty facile and easy. But really to build a liposome with a nice aqueous core, unilamellar bilayer, is really quite challenging. So, you know, liposomes were first described in the 1960s surrounding blood cells. And the first method for forming them came about in 1965. This was thin film rehydration. Since then, many different methods have come about, iterating on like the bulk formation methods of the thin film rehydration or electroformation. We have now chip-based methods or dice methods, which I'll get into in just a second. But you know, we can build them on a chip. We can build them in a column. The methodologies have certainly expanded, and they all have their kind of particular cases that they're suited for. I urge you to take a look at a review, but I'll be focusing primarily today on DICE liposomes, which stands for density mediated interface crossing encapsulation. And this kind of whole liposome formation system was initially developed in 2016 in the Keller group, and it's been kind of really expanded in the Granzinger lab as well, with eDice and kind of derivative systems. But in 2015, Morita et al. Published this center figure here, basically a liposome spin column that worked by extruding under centrifugal force an aqueous droplet from a glass capillary, that aqueous droplet being your lumen of interest in the ultimate liposome. This would pass through a lipid and oil solution, and then through a monolayer between that lipid and oil solution layer atop an aqueous collection volume. In 2017, another group looked at what would happen if you had the orifice of this kind of system submerged or in an endo conformation in the lipid and oil. And in 2022, we developed a 3D printed version to kind of avoid some of the more complicated manufacturing processes associated with the tight tolerances of the really rigid glass capillary. And this was our version 1 of the spin column. And this was in a 1.7 mil tube. Ultimately, we upgraded this to use an HPLC tube rather than a glass capillary. This gave us a little bit more repeatability and the ability to, you know, the HPLC tube can be ordered more easily. The next iteration was from version 2 to 3, where we changed from a 1.7 mil tube to a 5 mil tube, and this allowed for more optimal volumes in terms of formation. So we could, you know, load a maximum of 10 microliters into that glass capillary. That would be 10 microliters of the ultimate synthetic cell cytoplasm that you're trying to make. Obviously, the collection volume being 30 microliters, you're not getting a yield of 10 microliters of lumen. And so by going to the 5 milliliter tube, we get a little bit more collection volume, and the ratios of lumen to exogenous media work out a little bit better. There are also some theoretical considerations with this kind of liposome formation method apart from others. So compared to something like emulsion transfer or E-dice, where you're forming an emulsion, you're layering that emulsion over a monolayer that has maybe pre-incubated, and then under centrifugal force, you're driving the reverse micelles in that emulsion through the monolayer, kind of zipping on that outer bilayer to turn them into liposomes. As opposed to a system like that, this system where the micelle formation in the lipid and oils layer, and then the micelle propagation through the monolayer happens all in one step. And so the absorption of lipids to the aqueous droplet in the oil layer is kinetically limited. So, there are just different considerations to think about in terms of liposome formation. And this is I'm showing you here a schematic of the newest device version 3, where you see the bottom aqueous collection volume, the oil layer that the orifice protrudes down into, and then kind of a loading chamber where we load the lumen. So thinking a little bit about, can we better make liposomes by optimizing various processes that are going on during that liposome formation? So I mentioned that the emulsion formation step and the emulsion to liposome transition happens kind of all in one process. But just thinking about like separating that out, is there a way that we can optimize the reverse emulsion formation and then kind of get better liposomes? And so that's kind of what we did. So we primarily did this by looking at the choice of lipids. So there was a work in 2021 that showed that if you used a lipid that had a more optimal packing number, and lipid packing number relates to the size of the head group versus the size of the tails, and whether or not the lipids would be more suited to forming reverse micelles, that is inverted micelles, or regular micelles. We would want to be forming inverted micelles in that oil layer so that as it propagates through the monolayer, the outer bilayer can zip on. And this paper in 2021 basically found that including lipids that have a smaller head group, so that you have this inverted cone packing, helps with the liposome formation. And so we've started adding 30 mol% POPE to our lipid and oil solutions. And then additionally, in that same kind of vein, thinking about the process of liposome formation, where you have first absorption of lipids to this aqueous droplet to form a reverse micelle, and then propagation of that reverse micelle through a monolayer. If you're including cholesterol in your liposomes, the cholesterol is, assumedly binding to the unsaturations in the hydrocarbon chains of the lipids. And so this could be an additional step post absorption of lipids to that aqueous droplet. So the inclusion of cholesterol is also a potential benefit for increasing the stability of those protoliposomes, the reverse micelles. And then we've also been looking a lot at surfactant concentrations in choice. So the standard surfactant that's been used in liposome formation has an HLB that is a hydrophobic lipophilic balance of 13.4. This is not suited for reverse emulsion formation. And so by switching to things like SPAN 80, which has a HLB of 4.3, or ABLE WE09, which has an HLB of 5, we've been getting better liposome formation. Just by kind of trying to improve reverse micelle formation in that lipid and oil layer. And then thinking about the next step, how can we improve liposome formation by improving the process of the reverse micelle propagating through the monolayer, that is the bilayer maturation or the zipping of that outer bilayer. One of the things that was pointed out in literature, particularly with C-dice, was that the change in orifice size was not related to a difference in liposome size. by extruding into just an oil layer, not layered over an aqueous volume, we were able to kind of take out the emulsions and look at them under a microscope. And we found that by changing the orifice size, by changing the amount of surfactant present, by changing the amount of phospholipid present, we were able to tune the size of the droplets that were forming in that LIO. And if you're thinking about the droplets going through that monolayer, one of the things that's been noted in literature is that you have to have an incubation period where lipids absorb to that monolayer, which is to suggest that if there are no lipids at the monolayer, or they're depleted at the monolayer, the reverse micelle propagates through that oil-water interface, and there are no lipids to add on that outer leaflet, and you don't get a liposome. And in this context, one of the observations that we've made is that sometimes we're making droplets, intermediary droplets that are just too big, upwards of 800 microns. And if our 5 mil tube is, about a centimeter across, 10 millimeters across, think about a rock going into a body of water and the ripples that causes. There's a possibility that we are depleting the lipids in the monolayer by virtue of trying to force too large of protoliposomes through it. And in that sense, maybe the monolayer is acting kind of as this dynamic filter medium, where you're not controlling the size of the liposomes with the orifice that you're extruding into the oil layer, but you are controlling the size of the droplets that you're making in that oil layer, and the size of the droplets are disrupting the monolayer in such a way as to control the size of liposomes that you get. The other consideration too at the monolayer is something like a force balance. So you have a contact area between the reverse micelle and the monolayer, and as soon as you have that contact area, you have pressure across that. Not just due to the physical impact of the reverse micelle under centrifugal force against the monolayer, which induces some sort of bulk pressure. But also, if you have, for instance, a hypotonic or hypertonic solution, you have some flux across that contact area. And we think that this is ultimately why it's important to have either isotonic match between the lumen that you're trying to encapsulate and the exogenous buffer that's serving as a collection volume, or to some degree having that exogenous buffer be hypotonic, so that there's a little bit of back pressure and you don't have deflation at that crossing. And this is something that's been kind of noted by other people in literature as well. So kind of making these changes to the reverse micelle formation and the monolayer crossing, involved adding some surfactants to the system. And one of the things that we wanted to do is look at whether or not those surfactants inhibit TXTL. And kind of interestingly, we found that basically as you go higher in HOB, this is meaning that the surfactant is less likely to segregate into the water. This kind of makes sense. The surfactant is less inhibitory towards the TXTL. So the surfactant that we end up using in the lumen compared to the surfactant that is in the oil is quite high. It can dissolve into the aqueous volume of the lumen. And the surfactant that we use in the oil is quite low. This is referred to as a paired surfactant system. And in my conversations with some of the cosmetic engineers at Dow Chemical, one of the things that they relayed to me was that a paired surfactant system will always result in a smaller emulsion size. And so this is kind of good for us in the sense of trying to reduce that intermediary reverse micelle size in terms of monolayer propagation. And that's something that we're currently trying to tease out. The optimal concentration of surfactant inside the lumen and the optimal concentration of surfactant in the lipid and oil solution. And then just here, what I'm showing you is that the surfactants that are the higher HOB that would be in the lumen turn out to not really inhibit the TXTL reaction nearly as much as the hydrophobic lower HOB surfactants that would be found in the oil solution. So that works out well for us. In terms of building a mycoplasma cell, synthetic cell, or liposome, I've listed here in order of kind of difficulty what I think the issues are, right? So recapitulating the lipids of the mycoplasma, recapitulating the proteinaceous membrane pores and transporters on the membrane, and then obviously encapsulating the intact genome. Tyler talked a little bit about installing the lipids, but here part of our conversation is about, do we need to start with a minimal mycoplasma, a lipidome, as was kind of published by the Sainz lab, or can we work with a lipid composition that is more suited to liposome formation with my system, with the genome and the requisite membrane proteins installed, in such a way that over time the membrane would be remodeled and this would maybe be okay? Or do we need to start with a membrane that is at the onset more similar to the mycoplasma, in which case we're considering both making liposomes from lipids extracted straight from Syn 3.0 or other mycoplasma, as well as just recapitulating the minimal lipidome, which would involve the palmitic and the oleic acid in addition to cholesterol. I think another really important point with the lipids in the membrane is this question of feeding. So a lot of mycoplasma media will have lipids or fatty acids in them. And the issue is that the cholesterol and the palmitic acid, the oleic acid, these act like a detergent and would ultimately destabilize the liposomes at some point. And so we need to identify a tolerance of the liposomes to lipid feeding. And the way that we're kind of thinking about doing this is this calcium sensor called IPPS. Basically, we'll make liposomes that contain calcium cation, and then add in cholesterol, palmitic acid, oleic acid, and we should see a fluorescent shift from about 405 to about 480, indicating the calcium and the IPPS binding. This kind of might end up being a pretty clean experiment, just in the sense that we can take this initial reading, add in the palmitic acid or oleic acid or whatever lipid we're trying to feed, see some kind of leakage induced by indicated by fluorescent shift, and then do a Triton X100 lysis of all of the liposomes to kind of get the maximum fluorescent shift. And so then we have kind of a nice internal control to look at that. Another thing I'll point out too at this point is that some people have been looking at whether or not you can stabilize the lipid feedstock with cyclodextrins, so that they're less inhibitory or less disruptive to the liposomes. And that's something we would certainly kind of integrate into this analysis. So Tyler also talked about the installation of SecYUG and other membrane proteins. This is coming from the Gleason lab. But here, the protocol for installation broadly follows the schema of make liposomes, destabilize liposomes in the presence of the protein of interest. You have the protein then integrating into those destabilized or slightly destabilized liposomes. And then you're restabilizing the liposome by removing the detergent. And now you have your protein of interest in the liposome. And so we're looking at this for SecYUG, which is perhaps the most important of the membrane proteins that we're thinking of putting in the chicken and the egg context, just because SecYUG will help us install other membrane proteins that we synthesize and translate in situ. But then we're also looking at hemolysine. So liposomes are very fragile, and I'll get into that a little bit more. One of the reasons why they're so fragile is their sensitivity to osmotic conditions. Adding in the hemolysine might give us the ability to have more relaxation in that osmotic system, as well as the removal of waste products in such a way that would maybe benefit the actual transcription and translation reaction. So installing the Syn 3.0 genome, I think, is honestly going to be the hardest part. And I think the question is, does Syn 3.0 genome survive this extrusion? And so in terms of these different HPLC, we work with 25 micron, 50 micron, 75 micron, and 100 micron. And I believe it was tried by Andre passing the Syn 3.0 genome through a 50 micron tube, and it seemed to be somewhat intact. So Tyler has sent me a genome and mycoplasma to try, just extruding and see how that goes. But he touched on this a little bit. The kind of ways that we would solve this if this is an issue is, do we need to form one liposome population with extract and maybe install the proteins into that? And we would do that with this spin column system, which I forgot to mention earlier when I was talking about the iterations. We're calling this mini dice. I called the 1.7 mil system micro dice, and so when we've gone up to the 5 mil system, we've called it mini dice. But one way to get around whether or not the system would destroy the genome would be to form liposomes that have the extract in them, because the mini dice has better encapsulation efficiency and can give us liposomes of the size that we're more so interested in. And then forming another liposome population that actually contains the genome, maybe perhaps by thin film rehydration, if it doesn't survive the extrusion through the spin column, and then using controlled fusion to fuse those two liposome populations. I mean, obviously, it would be really ideal if we can just kind of do it all in one go, form the liposomes with the genome inside, form the liposomes with the extract inside, install the requisite membrane proteins, and go from there, with feeding the synthetic cells being kind of the only issue, but we'll have to see. And then I just kind of wanted to show you some results with the liposome system. So here on the left, I'm showing you two micrographs from earlier in the summer. Red is the membrane, and magenta is a bacterium that I am encapsulating inside the liposomes. And just to kind of further hammer the point home of how fragile the liposomes are, I'm showing you a very preliminary gel on the right of supernatant versus pellet of the liposomes. And you can see that there's not very much difference between supernatant and pellet, which is to say that working and handling the liposomes for distal assays, whether it be microscopy or whatever it is, they're very fragile. So there's a lot of consideration that has to go into that. But being able to build better liposomes not only allows us to do the synthetic cell stuff that we're talking about here today, but also kind of investigate some biology. Here I have a video of one of my bacteria inside of the liposome swimming, which I think is kind of fun. But yeah, I'll take questions. I'd like to acknowledge my lab, particularly Tyler Barton and Grace Gustafson, who are two undergrads working with me this past year. Yeah.
Thank you, Orion. What's your bet when you and Tyler get together? Kate says 50-50, it's going to work first time. What do you think?
We just need one to work. Sorry for cutting in, but it's an empirical thing. We just need a single cell to have a genome in it that can boot up. So we're going to try to make millions of liposomes and try to get one with the requisite materials needed. That's my opinion.
That's my opinion too. I think that it's a 50-50 bet in the sense, particularly that I think the biology of lumen chemistry is good. I think the engineering and physics of the liposomes is good, but obviously they're not perfect, and putting them together is certainly a complication. It would be very exciting to see.